Beth, is the botryoidal texture that you are calling out a function of the ferromanganese crusting?
Yes. Alteration of the underlying rock, or yeah, the precipitation of these mineral crusts on top of the rocks. Jess, can we have a look in this area for some of the rocks?
Yeah, let's do it. Is that a sea pen?
Yeah, another kind of sea pen there too, maybe.
Yeah, found us a tiny little pocket of sand to settle in.
That's what they do.
Okay,
Soft sediment experts.
lasers, yeah, great. Yeah, so one of these rocks might be suitable for my project. Okay, Rennie, if we want to maybe pause the ship's move so we have a little bit of time to spend
Bridge
here.
nav. Hold position.
And can we get a closer view of this little bundle of rocks right here?
Yeah, let's do it. Stand by 1 moment.
It's hard to say.
Yeah, if we had...
Yeah, Argus will swing. I can back up, but we might still have to.
Go ahead and push on in there, please.
Let me back up like 20 meters, maybe.
Roger. Jake, you want to get the arm out real quick?
Yep.
We don't have to... We can do a flyby. Any of these look appealing here, Beth?
Yeah, I think some of these will do. Let's maybe try for this one right in the middle. Looks like it might be the easiest to get.
All right, I'm backing up a little bit.
Roger.
So you want the one right on the lasers?
The one right in center screen there.
Yeah.
Roger that.
Go ahead and give it a grab, and if we miss that grab, we might to pull up and come back.
Okay.
Nice. I think that'll go. Very nice.
Oh, that's great.
Rounded.
Still all right on Argus. I'll just watch it.
Nice grab.
Yeah, that's great. We can just get a couple quick snaps of that.
Yep.
And then...
Go ahead and push on in there a bit.
Porch light.
Oh, yeah, Rennie, you're on it.
I don't know if it did anything.
Maybe. In spirit, it did.
This is going to go in the forward bio box, please.
Roger.
Good.
And you guys don't want a water sample here, correct?
Yes, if we have time.
Okay. Full wide.
Still okay in Argus. I'm keeping an eye on it.
Roger. I'll get you
Got it?
the
All right.
hydraulics here. Pulling out.
Does it matter which side?
Nope.
Yeah, just go to the easiest side here.
What's the sample number here, Steve?
109.
There you go.
09.
Go ahead and drop it there. Watch the camera. There you go.
Oh, it's a big one.
Nice.
A lot larger than I thought it was from a distance.
Good job.
All right, great. And then, yeah, let's try to get a water sample, and then we'll
Go ahead
get out of
and
here.
set up for the Niskin there, Jake.
Oh, yeah.
All open? Yeah.
All open.
Haven't done anything yet.
So we've done this a lot on our watch, but since we have quite a few viewers here today, this is a Niskin bottle mounted to the side
triggered by these little lanyards that are each color coded. And when the lanyard is triggered, the bottle will close, capturing the water that's just in this area, and it won't be mixing for the rest of the dive. That water can be filtered for DNA that happens
Still
to
good
be
on Argus.
water.
Reg.
Sub-bottom's all right.
Jeff, can we look at the pilot top-down view from channel 3, just so our
Triggered.
audience can see Jake using the manipulator?
Is that number 6?
Yeah, it was the
Alright.
white lanyard. I can tell what number that was.
White lanyard number
I
6.
can use that view too, but then I'm in it. But
All right. That's a fun view. Haven't seen that one before.
All right, yeah, log those 2 things. We're picking up. I'm going to
Okay.
catch up a little bit.
Great.
And then, do you want me to keep moving along our track or kind of hold here for a little?
So I think what I was discussing with the other co-lead scientist a few moments ago was, since the density here is relatively sparse compared to what we have seen on other dives, that we might try a strategy similar to what we did on the last dive, where we
Nice job, Jake.
put in ship's moves that are maybe a little bit faster, where we're moving through the water a bit faster,
Can do.
until we see maybe some higher density, and then we can decide to drop a point and come back, just so that we can make some more distance.
Okay,
Does that sound good, Rennie?
sounds good.
Yeah, let's do it.
All right, let's go 0.4 for a little bit, see how that feels.
Point
Yeah.
4 sounds great.
And so for our
Uh,
audience at home, we'll be
245.
245, Reg.
moving through the sea floor,
Zipping along a
moving
little
over the
more.
seafloor a bit faster. We won't have a lot of time to do some of these zooms to get closer views, but it gives us a way to see the seafloor a little bit faster and then
Cover some
slow
ground.
down if we want to. Yeah.
Steve, how much water do those Niskin bottles hold? Maybe Beth knows too, I don't know.
I believe they're 5-liter capacity.
Yep, that's right.
Each one is 5 liters.
Cool, thanks.
So from every sample, we collect 1 liter for an ongoing eDNA study. And then for the samples that
Sure
are
thing, Rennie.
co-registered with where we pick up rocks for the microbiology experiments, then I'll collect the remaining water for filtering through a filter with a smaller mesh size than what's used for the eDNA study to specifically capture more of the microbial end of the spectrum, so bacteria, archaea, even some giant viruses. And yeah, that's what we do.
How common are viruses on the seafloor?
Viruses are everywhere in the ocean. In a rough rule of thumb is that for every bacterial cell, there are 10 viral particles. So they have kind of a 10-to-1 ratio. But the study of marine virology is still somewhat new, so there's lots to still be learned. And most of the viral particles will actually go through the filter that I'm using. They're smaller than the pore size. So it's only going to be phage that are incorporated into cells, infected in cells, prophage that have basically viral DNA within the genomes of these cells, or viruses attached to cells, or some viral particles that are actually larger than the pore size and can be captured.
Have there been any documented zoonotic effects from marine viruses?
What do you mean?
Like transmitting to humans?
No. There are no known incidences of that.
Yeah, I think so. Yeah, there was a question in the mess that I was having with someone about fungi yesterday.
Yeah.
Is there a good diversity of deep water fungi, and are they responsible for similar biogeochemical roles here in the deep sea?
Yes, yes, and yes. So there are many different species of fungi that are found in deep sea sediments in the water, and increasingly evidence for them in the rocks as well. There's even some suggestion that kind of pore structure in the rocks can be formed by fungal hyphae kind of moving through the rocks, breaking open new spaces. There's definitely some research in that area. Yeah, and it's thought that an ecosystem role that fungi play is to break down larger molecules of organic matter into smaller, more manageable pieces of organic matter that other forms of life can use, which is a similar role that they play, for instance, in soils on land, where they break down and decompose organic material.
Interesting structure in Argus view.
Yeah, you can see how this is really just like a spine of a ridge that we're on top of, which is again how it looked in the HYPACK view. Looked like a really, really narrow ridge.
Sharp ridge, yeah. What does it mean that we're in a rift zone?
That's maybe, so I don't know that we're actually in a rift zone. Usually rift zones are more used for describing areas of the seafloor where you actually have like tectonic forces, subduction, other kinds of things that are pulling the plates apart. So the plates are rifting apart, and you have formation of volcanic ridges. We're in a hotspot type setting, we think. And so these ridge flanks that we're looking at, these are more like an ancient volcano, and it has these ridge structures. So they're not necessarily rifts, they're what I would call a ridge.
Starting to pick up speed with Argus.
So again, for our viewers at home, our strategy right now with this dive and our three-body system of ship to Argus to Hercules is that we've called in a move to the ship to move in a southwestern direction at a little bit faster speed than we have been going, which will pull Argus along also at a faster speed, and so Hercules will be driving forward to keep up. So we're going to see the seafloor start to go by and a faster speed than we have been viewing on this dive previously, to try to cover some more ground more quickly, and see if we get into areas with higher animal density before we slow down again.
Is it cold down there, Beth?
Yes, the deep sea is cold. Our temperature right now is hovering around 2.5 degrees Celsius, which is a little less than 40 degrees Fahrenheit. And so about the temperature of your refrigerator if it's on its cold setting. And that's a pretty normal temperature for most of the deep sea.
It's warmer where we are right now than back home.
Yeah, for those of us who live in New England in a normal basis, this feels like a quite cozy temperature.
Yeah, we haven't had any midday recoveries that I can remember, but it's kind of refreshing when you're out there in the heat of the day. Stick your hand in the box, and it's usually
ice cold.
Ice cold, yeah.
Can't have it in there too long or your hands get numb.
Yep. One of the rituals of diving in Alvin, I'm not sure if they do it anymore, but after you get out of being on Alvin, it's quite hot on the surface. And so you get kind of sweaty when you get out of the of the sub, they'll usually dump a big ice cold bucket of water on top of you to, celebrate your first dive.
Aw.
They still sometimes do that,
Yeah.
but they don't throw it at you.
Yeah. Yeah.
You
It's
sit down
voluntary,
and
yeah.
you are ceremoniously anointed with cold water.
Have we ever found fossils down here?
Yes. Well, not here on Tamana Seamount, because this is the first time we're diving on it, and I don't believe it's ever been dredged before either. So to be determined. These sands that you're seeing, these light tan spots, will have little tiny fossil tests of some microscopic life. On a previous dive at one of the other seamounts in this area, we did observe a mineral encrusted bone from a beaked whale. Fossilized skeletal remain of a marine mammal. That's the only obvious fossil that I could think of. We've also seen many, dead sponges and things, but I wouldn't necessarily call those fossils. No mammoth tusks.
Sometimes you can find in areas where there's fairly high crust accumulation, you can find scleractinian stony coral skeletons, often are encrusted with iron manganese coating, and so those are sometimes considered fossils because it effectively seals the calcium carbonate into the
Prevents
crust.
it from dissolution.
Yeah,
Yeah.
and actually, lately they've been used as pretty reliable geochemical archives for examining past conditions, past oceanographic conditions.
Kalamehu, we've got some viewers noting on channel 3 that you're wearing a hoodie. Concerned, are you cold?
Thank you for your concern. It's just comfortable. I don't know. It does get a little chilly.
Also wondering if you live in Hawaii.
Yes, I do live in Hawaii. Particularly, I live on the island of Maui, in Lahaina, as well as Honokohau. So if anybody's out there listening from home, good morning, gange.
Crinoid there on the rock.
Yep, that's what it looks like.
Yeah, some crinoids. And it is important to keep our control van fairly cool. We have a lot of equipment in here, a lot of computers running, a lot of monitors generating. Well, the computers are generating some heat.
Yeah, Jeff, could you put up the view from the web interactions camera? Yeah, so folks at home can see all of these computer monitors that we are looking at and the associated electronics that are feeding those data streams. So we don't just have to keep ourselves at an appropriate temperature.
Oh yeah, nothing like what upstate New York is facing right now. I guess it is -6 degrees there.
Oh, pretty chilly.
Celsius or Fahrenheit?
Celsius.
Oh, yeah. So not quite as cold as -6 Fahrenheit, which is...
May your coffee be hot and your hoodie's just as cozy.
If you look at camera 3, that's actually it's the dark blob of stuff, but that's what's generating the vast majority of the heat here.
Exactly. Where are we over there?
So all of this up in here and that over there, they're big, big heaters. It's actually not that bad in here. It's about 70 degrees.
Is it really?
Yeah, 70 degrees, 60% relative humidity. But the fact that we're not moving, one, and two, we do move a lot of air. There's blowers up above my head. There's blowers right behind you. And that's just, you kind of have a gentle breeze on you all the time, and that's why you start to get cold if you've been sitting here for a while. And it's funny, if you look at everybody in the van, you know, Rennie's got a fleece and a hat on, Jess is in short sleeves.
Yeah, I think it just depends really on where you're sitting.
Yeah, where you're sitting.
Yeah, I've had a couple shivers this morning.
Part of it, yeah, you want to stay a little cold just so you stay awake.
Yeah, I definitely notice that when I stand up, I get colder. I'm feeling a different airflow.
Yeah, there's, in fact, if you look at it right now, you can see there's a little yellow streamer there.
Yeah.
I have a very warm door next to me.
Yeah, there.
It's nice.
Utilizing conduction.
Yeah.
So for folks tuning in, we are currently exploring in the Tamana Seamount. It's the first time we are diving on this feature. It's an ancient volcano that is also a flat top geo. We're at about 1,667 meters and rising as we move to the southeast. Surprisingly, compared to other dives or roughly these same depths, we're seeing very sparse animal communities. So we have put in a relatively fast ship's move for some, to try to cover some ground pretty quickly and see if we can identify areas where the animal density picks up.
Colin, I have a note for you. Aloha, are the deep sea ocean sea cucumbers called loli, like their reef cousins? Mahalo from Honolulu.
Mahalo for your question. I was actually looking at that too. I have a limited sort of knowledge of this, but looking at the online dictionary, we see loli is in fact a word for types of sea cucumbers. There's a few others. So veli, maihole, kunono, hulali, hulalilali, upalu, kuneuneu, unai, kaukamakai. I think the kaukama might be cucumber. Yeah. So there, sorry, like I said, so these are a few different words that we might have, and I think that I personally might need to do a little more research to be able to see if they are classified as ones from more shallow reef areas as opposed to the deep. Which actually brings up a good point about the function of the cultural working group, who recently had a presentation and a document talking about the nomenclature of some of the species associated with Papahanaumokuakea. And that's going to be everything from birds to some fish. And yeah, I think that during this expedition too, we've had the opportunity to see a lot of these deep sea corals, and I was looking into it for like general terminology for coral. And it's going to be koa most of the time, but you know, seeing how these species and stuff are, well, for me anyways, very new and stuff. So I'm wondering what the CWG and the nomenclature who we might be thinking about in terms of Hawaiian language terminology for all of these species as well. And I believe that one of our SCFs is in the works of that as well. So yeah, mahalo for the question.
Yeah, mahalo. And Jess, we have a question for you. Wow, and just for some context, we have well over 100 viewers around the world. We've got some new viewers just wondering, why are we seeing some green dots in the middle of our screen?
Ooh, yeah, good question, good observation. So the green dots are our scaling lasers, so we know how big things are underwater. Yeah, especially really helpful for not only us when we're sampling, but also for scientists that are post-processing the videos as well, and they might want to, sometimes we do like video transects, and we can do surveys of the different marine life down there. So the scaling lasers are used both for an operations standpoint and also for science.
And what is the scale?
10 centimeters.
Thanks.
Or
do you have to calibrate those regularly?
No, we don't necessarily calibrate them regularly.
They're kind of fixed.
Yeah,
they're fixed, yeah.
They have like a fixed mount, fixed angle, so we don't really have to worry about them sliding around.
Right on top of the camera, right?
Yeah, it's right on top of the camera.
So they go where we look.
They better go where we look.
We look where they go.
Yeah, all the above.
Yeah, and welcome and thanks to all of our online viewers for joining us in this exploration.
Kind of a follow-up laser question, and I don't know if anyone has experience with this, but are fish or animals ever attracted to the lasers like a cat?
I don't know that I've seen them attracted to the lasers versus just the lights of the ROV.
Yeah.
Has anybody seen that?
There have been some instances I've seen with fish, yeah, and especially as you move into the very shallows, they'll follow you around. But I think it's because they're more familiar of using, the visible light spectrum. But yeah, there were some instances in 2019 where we were diving quite shallow in the Pacific remote islands where some fishes would, follow the lasers around.
I thought I remember shark situation, but sharks with lasers, that's a little too on the nose.
Very Austin Powers of them.
Yes, the dots are 10 centimeters apart. And, oh, thank you. I guess on YouTube we have over 300 viewers, so currently active. If you're watching on YouTube and wondering how to submit your questions for the chat, that is on nautiluslive.org. There is a submit question feature next to a green dot.
And speaking of green dots, coming back to those lasers, for folks who don't think in metric units very often, 10 centimeters is roughly about 4 inches.
You can see the Argus sonar and also just the visual is kind of matching up with our bathy that this is a less steep slope as we get to the top here. And just let me know. I'm going to keep following that, Beth, but if you want to be on the steeper side, let me know.
We can stay on the shallower part for a bit and see what it looks like. And if it doesn't seem promising, then yeah, maybe we'll swing around to one side of the ridge. And the question is which side? Probably the west side, since it seemed that the current was coming from that angle.
Oh, look at that lovely sea pen.
Yeah, let's keep this kind of south-southwest trajectory for a little bit.
Sure.
What are we looking at there in the center of the screen, Steve?
Do a 5-second zoom there.
That's a plume.
Go ahead, Jeff.
Plume.
Yeah, well, there's a couple different types of sea pen there. Yeah, but this is I want to say it's in something in the Peninsulae family. It doesn't look at all like the Enthoptylum
All right, full wide, please.
we saw previously, but there's some more of those Protoptylum red stick pens as well. Yeah, that was quite a big sea pen.
Yeah, pretty.
That one looks a lot like what I think of as like shallow water sea pens.
Yeah, more traditional, I guess.
Thanks, Rennie.
Yeah, still got a bit of that 230, but eventual 210.
Peninsula.
Roger that.
Probably. Genus Peninsula.
Does the ROV have UV light, or has it ever played with UV lights?
We don't have it. The short answer is we don't have anything on it. No UV lights on currently, and I don't think in the past. Yeah, I'm not terribly certain, but I don't think so. Not in the past.
I can't think of an instance. There was that modem on the Abyss lander that was towards purple, but I don't know if there was UV.
Yeah.
Do you remember what the wavelength of that was?
imagining that question is about fluorescence, observing it. Beth, earlier you said that the area hadn't been dredged before, they think. What would dredging entail, and what does that have to do with fossils?
So prior to the ability to go down with ROVs and carefully select rocks, another method for rock collection from the deep sea is to use a rock dredge. It's basically a chain feature that drags some chains along the seafloor to bring up rocks that geologists and petrologists and volcanologists can look at to get an idea of the rock composition of the seafloor. Still a technique that's used today in some places, depending on the research questions. And so some seamounts in the Papahanaumokuakea Marine National Monument had been dredged many decades ago. So for instance, the Liliuokalani Ridge that we'll be visiting in April, one of the seamounts in that ridge chain was dredged several decades ago, and we had one data point to give us an indication of the potential age of that seamount. But I don't believe that this seamount has previously been dredged. And so in relation to fossils, as you're dredging, if you had a larger fossil bone or some of these mineralized coral fragments that Steve was mentioning earlier, those could also be recovered in the dredge. But again, I don't think any of that has previously been done on this seamount. Anybody can look for that information on the NCEI database. Steve, can you help me remember what NCEI stands for?
National Center for Environmental Information.
All right,
there you go.
already got the bingo card.
I would defer to the mappers on that because that's where all the mapping data is stored too.
Yeah. But so there are different layers in those data sets related to dredge data. So our viewers at home can check out those resources online and see what's known about this region.
Got about 20 questions come in the last 2 minutes, so I'm guessing some YouTube viewers found the chat.
Yeah,
All
they might have
right.
been typing in the comments on YouTube.
Welcome.
Discovered
Yeah, thanks for all your questions. I'll try to hand some. These are great questions. Oh my gosh. Someone says hi to Uncle Rennie too.
Hi to Uncle Rennie. Hey.
Okay.
Guessing that's Joseph and Serafino.
A broad question here, maybe for Steve. What makes some of the areas more dense with life than others?
A lot of variables. In this case, the one we've been talking about the most, I think, has been flow conditions. So if there's a steady flow that provides food particles, marine snow to suspension and filter feeding organisms, just as important is the stability of the substrate, particularly for long-lived deep water corals and sponges. If their substrate is not stable, they will not be able to grow very big or grow at all. So you typically won't find them on talus material like this. They're more associated with boulders and things that are more stable, and sediments, rocky substrates that are more cohesive. Like, great example coming up here. This little larger piece of rock seems to be supporting this large bamboo coral.
It's quite tall in Argus.
Yeah.
Rennie, can you zoom in on HYPACK, please?
Can do.
Thank you.
And then the final thing, among the top three that I think are really driving these patterns is larval supply. So
that's good.
If you don't find an organism in an area and it doesn't have an ability to release larvae and larvae to settle in an area due to hydrographic conditions around a seamount, you won't find that animal there.
Well, thank you. Yeah, and we are here on
there, Jake.
What was that?
Come up a little.
Yes, roger that.
And Rennie, I think given what we're seeing, if we can maybe adjust the ship's move to bring Herc and Argus just a little to the west side of this ridge.
Okay.
So that we can maybe see some steeper topography.
See pretty massive crinoid. Yeah, I can do that. So
all right, pull wide, please.
Let's see. I'll see where they are along in their move, and we'll go still in the southwest.
Yeah,
direction,
still moving southwest,
more.
but just get us kind of on the western side. Sure. See if that gives us any higher diversity than up here. Or higher abundance, I should say.
Yeah, and some folks are just tuning in, Beth, wondering what kind of seafloor substrate we're seeing right now, and is it possible to know how old it is?
So we are on an ancient volcano, and we're on a ridge coming out from that volcano in kind of a northeastern direction. What we're seeing are the underlying igneous materials that came out when that volcano was active. So in some cases we're seeing extrusive basalts, sheet flows, pillow basalts. They seem to be highly altered. So you can see the texture kind of looks kind of crumbly, what we sometimes refer to as like a botryoidal texture, with sediment that's also filling in the little gaps. And so those crumbly texture is a reflection of the mineral crusts that form on the outside of these rocks over time. Just by looking at the rocks, we cannot tell how old they are. That is a goal of this expedition, is actually to collect some of these rocks so we can try to date this seamount and the volcanic activity in this area. That's a bit of a tricky challenge because if these seamounts are very old, which we anticipate that they are, then the rocks may be highly altered. And so finding a rock that has fresh enough, unaltered mineral grains on the inside is a key objective, but it takes some finesse. If these seamounts are part of the genesis of the northern Hawaiian island chain, then they would probably be in the range of, let's say, 20 to 40 million years old. There are some thought that maybe these seamounts are part of a different hotspot feature, with the Hawaiian islands overprinted on top. And in that case, they would be quite a bit older, maybe by several tens of millions of years. So to be determined.
Exciting, and that analysis can't even take place on the ship, so we're talking about
correct
weeks to months till we have some answers there.
Yes, so the rocks that are collected on this expedition will first be sent to an archive where our shore-based and ship-based science teams will request splits of those rocks that will then be sectioned and separated in labs that have those kinds of technical abilities. And then there are specialized facilities for trying to get at these ages using what's called argon-argon dating, based on radioactive decay of materials in the original rocks.
Someone's predicting that we have collected nearly 400 pounds of rocks at this point, so there should be some good ones to date. Are these seamounts still active, Beth?
No, they are not. So all of the features we are diving on are ancient volcanoes. If we were to zoom out on HIPAC or bring up Fledermaus, we would see that actually the seamount we're diving on was formerly subaerial, above the sea surface, and then as it subsided, had a carbonate cap on top, and then it had erosion of a flat top surface. So that's definitely a good indication that this is an old, very old structure. There's no evidence of active volcanism. There's also, we haven't seen any evidence of hydrothermal fluid flow, even at relatively low temperatures, some of the indicators that we might see for that. So there may still be some fluid circulation in and out of these seamounts, but not to a degree that we're seeing strong indicators of it in temperature.
Thanks for all your questions. I'm trying to move through them here. I know we've talked about this a lot on our watch because we're...
Where's that?
We got our first tunicate sighting.
Yeah. Tunicate hunters.
All right.
Oh, there you go.
Yeah. There we go in the screen.
We were just talking about this yesterday.
Yeah.
Go ahead and do a quick partial.
How do we spell tunicate?
Tunicate with no silent C's or P's.
Yeah.
Very cool.
Very cool.
Good eye, Jess.
Yeah, what are those
All right, full wide, please.
colored granules? I'm not sure what's
Yeah.
on the outside of that. Sometimes they have gastropods associated with them, so snails will attach to them and graze, presumably on something or on them themselves. Megalodicopia genus.
And I have to say it again because it's so cool, but in the larval stage, tunicates have a notochord.
Very nice. Zoroaster, big sea star.
So that makes us related to tunicates. Is that what you mean by that, Lindsay?
Yeah, they're our closest invertebrate cousins.
I definitely feel like a tunicate after I eat a big meal, so
Yeah.
just I can see it.
Just write in when you most feel like a tunicate, audience.
That would be a good quiz.
Lindsay.
Thanks, Renato.
You were thinking of some trivia or quiz questions, so maybe we could have a tunicate themed.
Yeah.
Little fish or something.
Little fish.
Yeah, very small fish.
Team name suggestion, highly altered tunicate hunters.
Oh.
We've got some long-term listeners.
That is abbreviation as HATH.
Oh,
Ah.
What if we went with mildly altered?
mildly altered.
Moderately altered? Massively altered?
Jake, how deep can the ROVs go?
Just about 4,000 meters.
Just about.
Are these nodules,
That
or are
would be
they
the edge.
talus?
Yeah, I actually think it's more like a botryoidal texture of the underlying material, and then the sediment around it makes it look like a nodule. But I think that's all actually intact.
Got it.
Is that another tunicate there on the left?
It is.
So a little bit further to get us over.
360, Raj.
260.
Oh, 26, Raj.
I think it happened again.
Oh, nice. At least you know the fix this time.
Very round boulder here.
It's a little interesting field.
Yeah, so these look more like some nodules up here. And we're seeing that kind of sediment drift off ahead. So heads up to our camera operators. If you see any ripples, take some pictures for our science party that's interested in that. Doesn't look very ripply.
Sea pens in the nodule field.
I come up on the delta here, Jake.
This little disconnect button under the
There's a
the
little
bottom.
shrimp
There you go,
photobomb.
that disconnected.
Yeah, so looks like we're approaching a tailless field here. Material that's slid down from somewhere else.
I'm thinking just looking at Argus sonar, we might be kind of approaching the edge of this top. Might fall off after that dark spot at the top.
Yeah.
Yeah, let's see what we find.
So just inching over there. Is it all?
Interestingly, the oxygen concentration is coming back up as we're approaching what may be a ridge drop-off. You can see that kind of information if you are on the Nautilus Live website. There's a little data bar on the right side showing some of Hercules data, and if you click on the More Data button or link, you can see some of the real-time data streams that are being collected by sensors that are attached to both Hercules and Argus.
Yeah, and the reason we're not slowing down and taking long zoomed in looks is that we're really looking and interested in areas of higher diversity and abundance here, or density.
Yeah, correct. We purposefully put in a relatively fast ship move so that we can cover a lot of ground more quickly. So we're not taking our time and being deliberate on imaging right now in an attempt to try to find spots with higher abundance. And then once we find those, mark them and spend some more time there.
Someone's surprised that we haven't seen more plastic waste, but it is important to note that this is an area that's not heavily trafficked by ship traffic or recreational use. Not to say that waste can't end up here from anywhere else, but, and we have seen a few examples on our dives in the monument. I think the largest being a bit of derelict fishing gear.
That might be a good trivia question for later. See who's really been watching this whole time. What was the first piece of foreign debris that we saw on one of the seamounts in our early dives?
Let's see. People in the van are not eligible for this trivia question.
Darn. Just wrote up that
I just
report
had to double
too.
check.
But I am proud that you know the answers.
You've been paying attention on our watch. Was it the 7-Up can? Yes, it was. Ding, ding, ding.
Nice.
Yeah, and a very
Correct.
old one, right?
That's right. Someone dated it to the 1976 to 1978, somewhere in there, based on the design. Is it possible to get an estimate of the sediment ponds from the Argus sub-bottom profiler? Do you see that, Jake, in the sub-bottom profiler?
Oh, so we're troubleshooting a
Gotcha.
GUI.
What was that about the sub-bottom?
Oh, no worries. You're good.
An estimate of these sediment pond dimensions. So sub-bottom really shows us it's like one reference point, the Z.
Yeah, you can. So
Okay.
the bottom, I don't know if you can't see it, it's not on the screen, but the low frequency data display, that's what kind of transmits through the sediment, and there's ways of looking at the returns in that to determine what your sediment type is in the seafloor, but I don't know off the top of my head or have done much sub-bottom analysis, so couldn't tell you, but it's basically looking at the intensity of the returns in the seafloor.
Better. All right, you ready, Jake?
Sure.
Beth, I'm just bringing us
Pilot
over the other
change.
side of this so then we can kind of face in to the
Yeah,
slope.
it's exactly what I was thinking.
Yeah.
If we could maybe come over it and change our heading a little bit to kind of just track along.
And then we'll come down this way once we're oriented.
Yeah, exactly.
Yeah, cool.
See if doing some contour following, what that looks like.
So yeah, for people at home, we're just getting Argus, because we have this two-body system, we ideally wouldn't want to put Argus on top of a slope and have Hercules turned around and lower. It doesn't give us a lot of, gives us less depth for Argus to work with.
More risk.
Yeah, more risk in those heaves.
Yeah, so we want to try to get Argus well on the other side of this feature. Already we're starting to see some larger sponges on this ridge slope. So currently Hercules is facing in a westerly direction. Jeff, if you could bring up HIPAC in channel 3 so our audience can follow along. Right, so here you can see where the lines are farther apart is kind of the top of the ridge where it's a little bit flatter, and where the lines are closer together indicates steeper slopes. Each of these contour lines, I believe, is about a 10-meter difference, or about 30 feet. And the green icon indicates the ship, Nautilus, that we are on. And the kind of purple icon, pink icon, is indicating the Hercules vehicle and its trajectory. And you can see the black kind of compass line is indicating its heading. And so you can see we've come over to the western side of this ridge feature. And our intent is to kind of have a southwesterly move, kind of follow along these contours that are on the steeper slope. Because in some cases we see higher animal diversity when we see these steeper slopes. And we also decided to go to the western side because it seemed that the prevailing current when we landed was coming from that direction. And we can kind of see that a little bit now in the Hercules view. You see the particles in the water moving from the right side of the screen to the left side of the screen. So they're coming from the west, moving towards the east. And so that might be an indication that food particles to feed these animals are coming from that side. So once we get settled in with Argus well on the other side of this steep ridge, then we'll start moving along. So we can already see that there's less sediment drape on these rocks compared to what we had been looking at. Again, an indication that the currents are coming up over this ridge, and then maybe they're slowing down in intensity and dropping some of those sediment particles out of the water onto the rocks that we just previously were exploring. You can also see that the water turbidity seems to have gone up a bit. It's, the water is murkier.
So just going another little step to get Argus on the other side.
Yep.
And then we can track along. We'll start out, I think we're okay at 0.4, but if we're finding that just navigating the terrain, we can slow it down. Or if you're interested in kind of the animals and the rocks, we can keep it slow.
Yeah, maybe we start at 0.2 just to have a little margin of safety in this. And then if it still seems to be sparse, we can pick it up.
Yeah, that's the smart way to do it. Let's start off dangerous and then and then dial it back a bit.
And
maybe while we wait,
we also had
sponges.
Our ROV pilots switch seats. So we now have Jake driving Hercules and Jess driving Argus.
Already anticipate a 225 move. Right.
Can we get a partial zoom on that sponge while we're staring at it?
Yeah, we have a second to do that. Good zoom there, Jeff.
So a couple brittle stars attached to this rather large sponge. You can see that those green dots, the lasers, are roughly 10 centimeter, or they are 10 centimeters, roughly 4 inches apart, just to give you a scale for this sponge that we're looking at. A couple corals on the rocks behind it. Thank you, video, for that zoom.
Come by there, Jeff.
So
Yeah, let's get
you're in
that
a good spot, right? So you can see
spot.
yourself in down cam.
Yep.
So you can lateral along this feature, always facing the feature.
Okay.
And then, yeah. So go ahead and try lateraling over to the right.
Beth, can sunlight penetrate the steep?
No, it cannot. We are at roughly, we're a little over 1,600 meters below the sea surface. So multiply that times 3, a little over 4,000 feet. Water, sunlight dissipates in seawater within a few hundred meters. Different wavelengths are absorbed faster than others. So reds, yellows don't penetrate as deeply as blue wavelengths. But yeah, so down here, if we were to turn the lights off, it would be completely dark. Nice. So the corals that we're seeing, calling out, these are not corals with photosynthetic symbionts.
Yeah, I think once Jake gets out a bit in front, I think it'll be good. Yeah, I think, yeah, because we'll be riding along that ridge, and if anything, we're going to get a little bit deeper. Sure thing. Thanks, Rennie.
So for folks watching along, we're still waiting for our 3 vehicle arrangement to get oriented to enable safe transiting. So moving the ship a little further out to bring Argus a little further away from the wall. So we have a nice margin of safety, and then we'll start moving along.
Sorry, so stay closer to the wall for visuals as we, and that may be lower or higher depending on.
Really high turbidity in this water.
Yeah.
Yeah.
And we can see that the current must be quite substantial. The particles are speeding by from right to left.
Giving you a tug here.
Oh, I see what's happened here. We've lost DVL because we're on a thing, so now we have to go rely on USBL.
Yep.
Is that still a sore subject?
Yeah.
Okay.
A little bit.
All right.
Yeah, I'm going to try to troubleshoot that after we are off watch.
Back towards Herc a little bit there, Jake.
Okay.
So that we don't give a tug, we get some.
So I think you're over here. If you look, you want to come back.
There you go. Nice. Come left and a little bit just aft.